Our project is basically based on the fact that astronauts lose a lot of bone density whenever they go into space. So when they come back, they lose about 1.6% of their bone density for every month they've been in space. When they come back, their bones are weak, and it's hard for them to move, and it takes about twice as long for them to recover from that. So our experiment, we sent up mouse bone cells onto the space shuttle, the last space shuttle, mind you, and we put in their vial a nutrient broth, dissolved oxygen, and human growth hormone.
So the brain is what says, or is what tells the body, let's grow bone, let's not grow bone. When you're in space, you don't have pressure on your body from gravity, so your brain shuts off that process.
So we actually have an actual picture of our experiment right there that we got from the UMJ laboratory. The bone cells need some sort of attachment, and in this case they attach to the bottom of the vial. So that was kind of our experiment to see how they'd be affected by microgravity.
They both form the matrix, which is what we were looking for. We just don't know if they grew faster or not because we used a human growth hormone. So we don't know if they grew along the same pace, but they both ended the same way. The whole point of the experiment was to see, is it possible for bone cells to grow in space? And the answer now is yes.
Instead of growing plants, I decided to go ahead and grow worms.
Okay.
This is a particular strain of worm that I'm growing. This is just a mock-up, obviously, but the adult worm lives to be about this big.
Okay.
So it's microscopic. So I've sent this worm aboard the International Space Station. I sent it aboard the SpaceX Dragon, the world's first commercial spaceflight to the International Space Station.
Mm-hmm.
And it went on the space station for about 6 weeks, and the worms grew for about 3 weeks.
Okay.
They actually just landed in Kazakhstan yesterday. So over the next couple of days, I'll be able to analyze the worms.
Well, what do you think happened to them?
That is another interesting part about the experiment.
Okay.
We have no idea. The particular gene that has been linked to all of the mutations they've had in past spaceflights. Pretty much when you mutate this one gene in this worm, it lives for up to twice as long, just by deleting that one gene.
Okay.
And what they did was they looked at that in this worm, this 1 millimeter worm, they're like, oh, that's nice, you can change it in this worm, but does it actually do anything? And then, so they did the next thing, they deleted the gene in mice, and they looked at the mice and how long they lived, and what do you know, mice live longer. And they did the same thing in chimpanzees, and what do you know, chimpanzees live longer. What happens when you mutate this gene and then go into space? That was my original experiment. When I read all of this, I found out that that gene, the gene that when you mutate makes the worms live longer, is actually the gene that has been linked to a lot of mutations that they've seen in space before. What they are going to do is, what they've already done actually is activate the experiment. They've grown for 3 weeks back in Kazakhstan, and I'll be able to look at this, and I'll be able to look at the genome and figure out, I've gotten rid of that gene that makes it live for twice as long, and that gene that is related to all of the mutations. What happens? Like, what happens to the other 15 genes that are changed in space?
Nobody knows.
Nobody knows, and I'm hopefully going to be able to try and find that out.
The name of our experiment is the effect of the microgravity on the use of cactus mucilage for water purification. Basically, we decided to do this experiment using the Opuntia ficus indica, that it's a cactus. And this cactus it's going to be used to remove heavy metals from water.
So is this the cactus you actually, that's actually very cool. Prickly pear, very good.
It can be a possibility to contaminate water from the moon with chromium-6. And besides, it's very dangerous. Chromium-6 is even considered as a carcinogenic agent. So that's why we chose chromium-6.
This cactus has a pectin that actually is the one that it's actually binding with the chromium with a ionic reaction. This reaction is because it has calcium +2. So this is a displacement reaction.
The chromium displaces the calcium, and the pectin gets a new layer of chromium-6. And actually, when this happens, the molecule gets heavier, and they sediment to the bottom, leaving the upper part water clean from chromium-6. That's how it works. But actually, we are not we're just testing the chromium binding process because that flocculation process that I just mentioned will not work in space. The pectin will not sediment to the bottom.
Right.
So what we did, we just try to find out a way to separate out the mucilage. So we just compare the filtration and centrifugation, and we find out that filtration was the best method to separate the mucilage bound to chromium in the solution.
And what do you expect to be the result of the microgravity experiments? And what do you expect to see once you get to analyze the samples?
We expect that the purification of water could be possible on moon surface. We decided to use chromium-6 because in the production of steel, right now they are trying to see if it's possible the extraction of steel from moon. There could be a chromium-6 contamination. So this could be a way to help that, to avoid the contamination and to get the water from moon clean.
So what you really want to show is that there's no diminishing effect from microgravity for this, so it's a very viable way to do it. Excellent. So let's hope you get good results, and very soon too, right?
Yes.
All right.
Thank you.
How did you come up with this particular creature?
It was on the approved items list on that,
Okay.
so we
And
thought it would be.
what's so special about it that makes it suitable for the experiment?
Extremely difficult to kill.
Yeah.
Okay, that's good to know.
And the other thing is it's eutelic. And eutelic means that when it reaches adulthood, it has a specific number of cells.
Yeah.
And when it grows because the cells grow.
Okay, it doesn't add more
It doesn't.
cells.
Yeah,
Okay,
it doesn't
I
add
get
more
you.
cells.
Yeah.
And so we thought that if we were to try to measure the size of cells in space, that would be very difficult because
right.
anything we would measure would be very minuscule.
Right.
But if we sent these things up in space,
Yeah.
if they were to grow,
Right.
then it's because of all of their cells grow.
Right, I get you. Yeah.
So it'd be easier to measure.
So did they survive the trip?
Yes.
Okay. Says that you couldn't really get a conclusion, is that right, or what?
Well, any change that took place was very minuscule.
Yeah.
That's kind of a cool experiment. Thanks a lot. Yeah.
Thank you.
What is a killifish?
It's a small freshwater fish found around Europe and Asia. Grows about 9 millimeters in its lifespan. It feeds off brine shrimp in the normal natural habitat.
How'd you come up with the idea to study the killifish and the bone density and muscle mass density?
Well, we wanted to figure out something that we could put in the tube that could be alive, yet small enough that it could fit and live in the tube. And we found that killifish can live in the tube and that we can see organs in the fish as muscles, bones, and kidneys. So we can test relationships like kidney stones and then bone and muscle mass between fish on Earth and fish in space to see differences.
So now, when you get results back and you find out that bone density is affected by the microgravity, how are you going to apply that knowledge?
We can then take the killifish and we can use modern medication and try to develop new medication, because the same thing happens to human beings as they go up and come back from space, because they lose bone and muscle mass as well. So we can take the killifish, use them sort of as test subjects for human medication.
Any ideas for improvement of your project? Or should you do it just like this again?
Try to figure out a way to send the fish up and maybe better hydration before they get mixed with water. The experiment came back on the lander that flew up with the Dragon here about a month ago, and it landed back in the last couple days, and our experiment is in Houston, being ready to be shipped Thursday afternoon.
And we'll take it up to Buena Vista University to do further testing to see our results.
Can you tell me a little bit about your experiment today?
Sure. Our experiment is the effect of microgravity have on the reproduction of E. coli 0157H7 and on the external structures known as curly. So,
where did you come up with that idea?
Well, we had like a limited amount of choices, so our teacher suggested E. coli and we went for it.
This bacteria was like unknown.
Did you have to do a lot of research for this project?
We had to do plenty of research. And we worked with a professor from Indiana University Northwest because the E. coli was very dangerous and disease-causing, so you had to be over 18 to work with it.
And what is your hypothesis?
Well, we hypothesized that the curly on the mutant strain would produce quickly.
Because the mutant in space produces really slow, so with that in a different place, it might change the reproduction time.
Yeah.
So it might might produce faster or slower. So we don't know yet. We send our experiment up in space, and there was miscommunication. So they cracked the ampule wrong, so they bring it down and take it up to space again.
Thank you.
Can you kind of tell me what the experiment was?
Well, we decided to do the growth of Lactobacillus acidophilus in microgravity, and this is a bacteria that causes cavities on your teeth.
Well, Victoria Holbert, who's not here right now, and myself, we're both in dental assisting at Monty Tech. So what we decided to do was, we heard of this bacteria before in class, so we saw that it was on the list of different materials you could use in the space project. So we're in the dental assisting program, might as well do something that relates to that. So we began to go into looking more into the lactobacillus and really what it does to the teeth, and we saw that it was an acidophilus. So we knew that once we looked into it, we saw that it actually erodes the enamel on the teeth and it causes dental cavities that way. So we figured this is a great thing that we could do to not only have a great experiment in space, but also just knowledge that's going to help us in later on in the field of dentistry.
Do you think you can use this in your dental classes?
I think I would definitely use this in dental school. Eventually be able to tell people that I had a project that went into space and I was able to learn about how this bacteria affected the teeth. I think it will really, really help out and be an impressive thing to the class.
That's great. Well, thank you very much.
Thank you.
What was your experiment about?
Our experiment is the effect of microgravity on the growth rate and pH of the Lactobacillus acidophilus bacteria. We're testing to see if the Lactobacillus acidophilus growth rate increases or decreases in microgravity. And this bacteria is found in the human intestines. So we're looking to see how we can better the health of our astronauts in space.
What does lactobacillus do for astronauts?
It's found in the human intestine, so it aids in giving a proper digestion, which helps improve overall health, removing chronic fatigue disorders, skin disorders, and preventing dehydration that causes muscle and joint pain.
What will you be measuring of the lactobacillus?
We'll be measuring the pH, since we know that the bacteria is an acid lover and it grows faster in an acid environment. We'll be checking to see the pH when it comes back from the ISS to see if it's a difference between here on gravity and in microgravity.
We'll be also measuring the amount of lactobacillus in each experiment to see if lactobacillus grows in a faster rate or lower rate in microgravity.
If the population goes down, then that means that we should give supplemental probiotics to the astronauts in order to aid in their digestion.
How do you get probiotics? What would you give an astronaut for probiotics?
We're thinking maybe powdered milk, fermented dairy products, and the pills, probiotics.
Excellent. Very, very good.
So why did you choose habanero chili as your experiment?
Living in a Mexico border town, habanero chili is one of our main daily dishes that we like to utilize. It's really popular.
It is a part of our daily lives and has been in menus for hundreds of years.
The reason we choose it is because astronauts reported that food tasted differently in microgravity, that flavors are dulled. So they crave food that is spicier or considerably sharp tasting.
The goal is to quantify the effect of microgravity on its chemical content.
This could provide astronauts the needed supply of endorphins to combat homesickness and the feeling of confinement. It also has vitamin C in it, so it helps your immune system.
This experiment can also lead to further studies on the effect of chili inhibiting the growth of bacteria, thus enhancing its medicinal value.
Exposure to microgravity for standard periods may not only affect the compound characteristics, but may also affect how the capsaicins bind with pain receptors.
What is frequency?
Frequency is how hot it is.
How did you test it?
We decided to use Scoville's method to test the habanero's piquancy. To get the habanero's piquancy, there's going to be 3 scientists who are going to test the habanero's piquancy. And we'll keep on adding the sugar solution until there's no longer piquancy left. The more sugar solution added, the higher the piquancy.
Did you enjoy doing your experiment?
Very much. We actually learned how to work as a team, and aside of that, we also became really close friends.
Our project is about whether or not microgravity affects how bacteria resist antibiotics. This is a pretty big topic because as the space program gets bigger, we have to ensure the safety of our astronauts in space, and that means preventing diseases and bacteria.
On the Apollo 13 mission, astronaut Fred Hayes was infected with this bacteria because it was in the water compartment. If you ever saw the movie Apollo 13 with Tom Hanks, the guy who was coughing, that was him. He was infected by the bug we chose. So it's one of the main reasons because, it has a tie to, history in the space program. Our experiment is taking 2 cultures of those bacteria known as P. aeruginosa. So we send one culture to grow in space in microgravity conditions, and we have another one growing on normal Earth conditions. And then when the space one comes back down, we test each culture with a series of antibiotics to see if it has the bacteria growing in microgravity creates more resistance or less resistance in the bacteria.
Our hypothesis is basically that microgravity will have an effect on resistance. We didn't really specify, we didn't make a hypothesis really on which way it will affect it. Basically, just expect a change.
Previous research has shown that the amount of time that a bacteria is in space will actually affect whether or not it becomes less resistant or more resistant.
What is the hardest part of your experiment, most challenging part?
I think the most challenging part was just finding the bacteria and choosing which one, because there are such a myriad of bacteria out there. We had to just find and select the right one, and this one was not that well known, but it's still very virulent. So I thought that was the hardest part for me personally.
One of the reasons why picking this bacteria was hard was because this bacteria is actually, I won't say dangerous, but it is dangerous to people with compromised immune systems. So our high school couldn't actually buy this bacteria. That's why we had Oakcrest help us. And their lab actually let us use this bacteria inside our experiment. And that was probably the toughest part.
Our hypothesis was that if we send it up to outer space, then the gravity will cause a slower rate of cell division.
Microgravity could cause the disorganization of microtubules, which is essential when the cells divide. It could also cause the chromatin, which is what forms chromosomes, your DNA basically, to condense at a less constant rate. So it wouldn't condense as fast, and as a result, we wouldn't be able to see the formation of chromosomes, and they wouldn't divide as fast.
How can you make sure your environment is pristine so your bacteria can grow without any interference?
So what we did was we sterilized the environment that it was growing in. We made sure that, for example, the fetal bovine serum and the EMEM, the Eagle's minimum essential medium, was really clean. And then we put it in sterilized flasks and grew it at room temperature in a controlled environment and put it in an incubator, which itself is controlled. So no bacteria was able to get in and there was no interference.
So if your hypothesis were true, can humankind benefit from this discovery?
Well, since we use fibroblast cells, fibroblasts are essential in the healing of wounds. So if our experiment were true, it would prove that, for example, if astronauts were to get hurt in outer space, it would take a longer time to heal, and so we might need extra medicine or other stuff like that.
Now, why are we concerned about putting oyster mushrooms or really any kind of fungus in space?
Well,
On Earth, there's a variety of scientists that have spent their entire lives researching mycology, which is basically the study of fungi. On Earth, they have practical uses as a biodegradable, renewable resource. They've actually created packaging peanuts out of the fungi mycelium, which is the roots, and it's biodegradable. They've created drywall and insulation that they actually put in some houses. And I know that in Europe, they've actually they've constructed bridges and other different types of traveling paths using them. When it's dried, mycelium is actually lighter and stronger than concrete.
Myceliums are prepared by just basically burning it and then compacting it together. So from that, there's no like fungi that's going to be growing from it because there's no like...
It's roasted mycelia?
Yeah.
Okay.
So it's roasted mycelium, and because that, there's no real nutritional value left in it anymore. So there's no fungi that's going to be growing from that.
Also, there's not a worry about bacteria eating it because fungi, even when they're dead, they're naturally antibacterial because they're the decomposers themselves, so they're pretty sturdy.
We did test for an optimal fungi species for our experiment. So we were actually testing different mushrooms, but what we ended up doing is we chose 3 very common and well-known mushrooms for this experiment, just for the fact that they're very easily attainable and they're popular to the world. So there's recent experience. There's been recent research done on those mushrooms because they're so popular and it's just safer.
So we've been cultivating these over thousands of years, so they're pretty much, we say stable, but you never know. Agaricus biturcus, it's the button mushroom. It's been cultivated in China since at least 1000 BC, and over thousands of years, they've selectively bred it based on the color and the shape. So now the mushrooms you get at the supermarket, they're not bitter. Most of them aren't toxic, we hope.
So you are not actually on a flight mission at this point, but you told me that you're still working on it. So are you going to try again?
Yeah, hopefully we'll be able to participate in Mission 3, and we might form an updated version of our proposal based on our ground truth.
All right then, well thank you very much.
So basically, we created a project and we fermented grape juice in space.
Okay.
We fermented it by mixing yeast with grape juice, and yeast breaks down the sugar molecules in the grape juice, and it puts off carbon dioxide, which is how we're going to measure the fermentation, and it creates ethyl alcohol, and the formalin is used so we can get exact timelines. Both of them can ferment the same amount of time.
What did you find out about your results on orbit?
Well, our results are still, we haven't been able to test them yet. They haven't shipped back yet. But we have tested our other ones. We've tested it for the ethyl alcohol levels. And for our next experiment, which it will be launching in October, we're going to use white grape juice so we can test it in a different way.
How did it taste?
Well, because there's formalin in it, you can't drink it because
it'll kill you.
But this is more for like testing like the scientifical purposes.
Oh, okay.
Yeah. This is something easy that you can test with like.
There's only a couple ingredients that we had to use.
The one going in space, there's a tube with grape juice in it, and then there's 2 smaller glass tubes within there, and one has yeast, one has formalin. And first you break the yeast one, and it starts to ferment, and then you break the formalin when you want it to stop, so we can have them like ferment the same amount of time, so we can get accurate results to see which one fermented more. Obviously, wine isn't the only thing that ferments. There's many other things that ferment. There's food that ferments. There's
soy sauce.
Yeah, there's
yogurt.
There's also medications and vaccines that need to ferment, and this could either make them stronger or make them ferment faster. And it could improve the medical field, improve the food field. It could do a lot.
Okay. Well, thank you very much.